All right, here we go. The context for the study should be familiar to you. As Allison has just told us, many promising HGM animal lines are under development. We've reached the point where we've had some early approvals, determinations of low risk, or determinations of non-regulated status for HGM animal lines. For example, here in the United States, after a safety review, our Food and Drug Administration made a low-risk determination for the marketing of products from beef cattle edited to knock out the prolactin receptor gene, leading us to a so-called slick coat and heightened ability to tolerate heat. Globally, as we just saw, there have been a handful of regulatory actions in Argentina, Brazil, Colombia, and Japan, allowing developers to go forward with production and marketing of slick cattle, a disease-resistant pig, and 3 fast-growing fishes with knockouts of myostatin or leptin receptor genes. With so many lines in development, other products presumably will be presented to the U.S. Food and Drug Administration in the United States or to regulators internationally. This raises the question of scientific uncertainties the regulators will face regarding animal health and consumer safety. And so this was the background for organization of a committee by the National Academies of Science, Engineering, and Medicine. With support from the U.S. Department of Health and Human Services, in which the Food and Drug Administration is embedded, and the National Institutes of Health, our charge is to identify methods for generating heritable genetic modifications in food animals, information on hazards to human health related to these products, information on hazards to the health of the target food animal species developed using heritable genetic modification techniques, information on other potential hazards that might reasonably be expected to occur based on relevant experience, for example, with genetically engineered crops, methods for the identification and analysis of intended and unintended genetic alterations, for example, off-target genome editing effects, insertional mutagenesis, introduction of antibiotic resistance genes that occur in the process of developing these animals with heritable genetic modifications, methods for the identification and analysis of potentially hazardous compositional changes in food animal products, challenges to the study of risks of food animals with heritable genetic modifications, and approaches to addressing challenges in study design, for example, appropriate animal models, long-term follow-up, and such, approaches to risk assessment in the face of uncertainty of the likelihood and magnitude of health hazards, and then lastly, to identify key knowledge gaps and to recommend areas of research for the near and medium term, which is defined as the 3 to 10-year time horizon that should be pursued by the National Institutes of Health, the Department of Health and Human Services, and other funders. And so the National Academies organized the Committee on Heritable Genetic Modifications in Food Animals. This committee includes 15 experts on animal breeding, biotechnology, food safety, and public administration, drawn from the academic and public research sectors. The committee was assembled last summer. After assessment of conflict of interest and public comment on committee membership, the committee was seated and approved last fall. We had our first meeting, a public meeting in October of last year, when we formally launched our study. We had a discussion with sponsors and agreed on the structure of the study. We held our first substantive workshop, Heritable Genetic Modification in Food Animals, Potential and Concerns, in Washington, D.C. in February. Most sessions were public. There were 25 presentations on topics ranging from methods for generating HGMs in food animals, traits that might be pursued within the next 2 years, views of stakeholders, animal welfare, genome annotation, biological mechanisms presenting novel hazards, novel resistance to disease, and genomics. And one session was closed. That was for committee deliberation. We have a report in press that should appear in September. Workshop 2 was held in Washington, D.C. in late July. There was an open session with 4 presentations on policy developments, on allergenicity, and on risk assessment. And there was a closed session for committee deliberation and work on our draft report. This is the structure of our draft report that we're working on actively at this time. I'll briefly describe the scope of each of these 5 chapters. Chapter 1 provides the technical background for the report and provides the context for our charge. We provide information on classical selective breeding and reproductive technologies, including artificial insemination, multiple ovulation and embryo transfer, and somatic cell nuclear transfer. We then provide background on the focal animal biotechnologies, that is, on gene transfer and genome editing, and on applications to mammals, poultry, and fishes. Having summarized the current state of animal biotechnology, we mention the first regulatory determinations. Chapter 2 regards biological mechanisms that may present novel hazards. We start by noting the benefits posed by HGM animals, including heightened production efficiency, improved disease resistance, and novel consumer qualities. We then turn to hazards posed by HGM animals, and these include the following: molecular genetic hazards such as on and off target effects and unanticipated metabolic outcomes. We discuss food safety hazards, including the possibility of altered food composition, novel or heightened allergenicity or food intolerance, and toxicity. We consider the hazard that disease-resistant HGM animals could become biological reservoirs for infectious disease. It could spill over into other animals, including humans. We consider the possibility of antibiotic resistance and mobilization of viruses from HGM animals used for xenotransplantation to humans, receiving those xenotransplants, and to people near them. And lastly, we consider the possibility of horizontal gene transfer. Chapter 3 puts these issues into the context of formal risk analysis, considering the likelihood of products of animal biotechnology presenting such hazards. The chapter starts by defining key concepts of risk analysis and then focuses on their application to HGM animals and food products. We present results of case studies and application of risk analysis to transgenic animals, including the gal-safe pig and aqua-advantaged salmon, and to genome-edited slick cattle. We have a long session on risk management, on how we can reduce risk to acceptable levels. We close by considering refinement of our risk assessment approaches through further research. Chapter 4 considers experimental strategies and methodologies to evaluate the safety of modified animals and their products. Among them, we consider methods for identifying molecular outcomes of our experiments, notably including identifying on and off-target editing events. We'll need to genotype founders and descendants in order to do so. We'll need breed-level reference reference genomes to do so effectively. Regulatory agencies are concerned with the possibility of compositional changes in animal-derived foods. We note that composition varies widely within and among particular animal products, and that we'll need much more baseline compositional data. We consider methods for assessing allergenicity and food intolerance. We distinguish between biological and statistical significance of differences between the products of conventionally bred and HGM animals. And we close by discussing incorporation of new findings into the risk assessment framework. In Chapter 5, we talk of scientific questions that may be addressed in directed research to be conducted over the 3 to 10 year time horizon. Among priority issues, we need to develop improved genome editing tools, for example, to reduce off-target edits. We'll need not only species, but also breed-specific genome sequences to effectively detect off-target edits. We'll need strengthened genomic annotation to assess metabolic and phenotypic effects of heritable genetic modifications. We'll need an improved food composition database to provide the baseline for comparison among foods from conventional and HGM animals. We'll need an enhanced understanding of food allergenicity and intolerance. We'll need to identify and manage factors affecting pathogen spillover from HGM or indeed any livestock into wild populations and into humans. We'll have to monitor viral dynamics in xenotransplant patients and those near them. To improve acceptance of HGM animals, we'll need well-designed communication and outreach strategies to reach consumers and the public. In terms of the process yet ahead, in late September, we'll have Workshop 3, which will be closed. The committee will meet to finalize the draft report. This fall, our draft report will go out for anonymous peer review. Over the winter, we'll revise our report and submit it to the National Academies for its approval. Our report will be published in spring 2025. With the first few HGM animal products now on the market, we expect more over the next few years. We hope that our report will have an impact on regulators, developers, and the public, so that HGM animal products that do enhance the sustainability of animal production will prove sustainable. Thank you, and Mark, back to you.